All right, so what I've been looking at is adaptations for drought and cold tolerance in eucalypts. So we know that plants have adapted to a variety of climates all around the world, including climates where water stress is particularly prevalent. And the ones I'm focusing on here are drought, where there's limited water available, and also cold, where freezing can create similar water stresses. And I'm looking particularly at eucalypts that have evolved to dominate Australian landscapes and inhabit pretty much all climatic niches across the country. So there are species exposed to extreme heat and drought, like you can see here, species exposed to frost and flooding, like these ones. And they're highly variable in their traits because they've adapted to such different conditions. So they vary quite a lot in things like tree height and leaf size, but also in their physiological function. And eucalypts are a particularly interesting study group because they're a clade of evergreen species. So where other clades have adapted by losing their leaves in times of stress, like through drought or over winter, eucalypts don't tend to do this. So they've had to adapt other strategies that have allowed them to thrive in some extreme climates that they've diverged into, particularly in terms of leaf traits, as they've had to maintain those leaves. And which is particularly interesting for me because a lot of what I'm looking at are leaf traits and hydraulic traits and how leaves have adapted to be less vulnerable to water loss or damage from water stress. And so while eucalypts inhabit climates all across Australia, I initially only looked at a small sample of Tasmanian species, as you can see on this phylogeny here, where we've mapped minimum temperature in the warmest month and precipitation in the driest quarter. So you can see all this red colouring here are those warm, dry climates, and the blue colouring is the cold and wet climates. And you can see over here the specific sites that I sampled each of these species from. And the main climatic extreme that I was able to capture down here in Tasmania was where species were exposed to freezing. And while I looked at heat and drought as well, there wasn't necessarily as much of that within Tasmania as some eucalypts experience on the mainland. So what I looked at across these Tasmanian eucalypts were several physiological leaf traits. So these ones here, and I took samples from 8 eucalypt species in the Symphyomertus subgenus and eventually supplemented that with data from 6 species from the Eucalyptus subgenus that Chris Blackman was looking at around the same time. And I was measuring a few traits related to cavitation vulnerability and cuticular conductance. So the first thing I looked at was cavitation vulnerability. And cavitation occurs when there isn't enough water in the surrounding environment for plants to continually replace the water they're losing through their leaves. So eventually the pressure pulling that water up from the soil and through the xylem will create an air bubble or a cavitation event. And this is particularly detrimental for plants because of how that halts water transport in those cavitated vessels, which will eventually cause plant death when enough of those xylem cavitate. And this can also occur through freezing, where the freezing and thawing of water in the xylem also results in air bubbles that stop water transport in a similar way. And so that's what I did here. I looked at how vulnerable each species was to cavitation. And to do that, I attached cameras or cavicams, you can see here, to a single leaf for each individual, where I got pictures every 5 minutes. And I also attached psychrometers to the stems so that I could consistently measure the water potential as or the pressure in the xylem as the branch was left to dehydrate in the lab. And what I got from that were a series of pictures that I could use to detect cavitation events, like the one that you can see here, which is a stylized version to show just the cavitation in the xylem. And from that, I created some vulnerability curves like this one. And so this basically represents all the different replicates of a single species. And what I was looking for here and what is represented by this line through the middle here is the P50, or the point at which 50% of cavitation had occurred or 50% of hydraulic function had been lost, which is basically an indication of how vulnerable these species are to cavitation events. And so that's what I'm looking at here, a comparison of that cavitation vulnerability among species. And so species with a lower or more negative P50, as you can see on the sides here, are more resistant to cavitation. And so what I looked at, so I looked at specifically temperature and aridity variables, because I was initially focusing on drought and heat. But I found that those two were highly correlated, at least across Tasmania and in the sites that I sampled from. So eventually we ended up combining those climate variables into PC1 that you can see here, using a principal component analysis. So on the left here, you can basically see the blue that's representing cooler and wetter climates, and the red over here that's representing warmer and drier climates. And this PC1 is the variable that I've used in analyses across my other traits as well. And so you can see here that, as we expected, species from warmer and drier climates were moving towards being more cavitation resistant. But what we also found was a trend towards cavitation resistance in cooler and wetter climates as well. And cavitation resistance has often been found more in correlation with dry climates, but we don't necessarily have those really hot, dry climates in Tasmania. So like you might find elsewhere on the mainland. But what we do have are sites that are particularly vulnerable to freezing. So, and that seems to be the main driver of cavitation resistance, at least in terms of Tasmanian species. And so that was just the overall trend, but you can see here how we break this up into the separate clades, where each subgenus seems to have evolved to cavitation resistance under a different set of climates. So subgenus Symphyomertus in the blue here seems to have become more cavitation resistant in those colder climates, so where they're subject to freezing. And then subgenus Eucalyptus has become more resistant to cavitation in those warmer and drier climates. And that's interesting because on the mainland, many Symphyomertus species have diverged into particularly arid climates, where here in Tassie, it seems to be the other way around. But it's also interesting that you can see something that's also interesting that you can see where most of the eucalyptus species in the red here fit this trend of becoming more cavitation resistant in those drier environments. Eucalyptus coccifera was a significant outlier and seems to be much more resistant than its climatic niche should suggest in terms of just that one subgenus. And it's enough that it's dragging down the relationship. And you can see here that fit without coccifera included. It's much tighter. And the reason that we're looking here instead at PC1 at minimum temperature is because when you look at the species that are exposed to freezing or temperatures below zero, you can see that Eucalyptus coccifera is the only species in subgenus Eucalyptus that's exposed to freezing. So it needs to be more resistant than perhaps other species in that subgenus. And so you can see there's kind of some crossover between the subgenera in terms of what climatic conditions they've adapted for. And so, so overall we found, so the next couple of traits that I was looking at were related to cuticular conductance. And so what I was looking at here was the minimum amount of water still being lost through the cuticle once the stomata had closed, and how stable that water loss was as the temperature increased, which comes down to a difference between the chemical composition of the cuticle and how permeable they are to water, which can differ between species. And so to do this, I used branches from the same trees as the ones I looked at for cavitation vulnerability. And I removed 7 smaller branchlets from each one, and I placed them in a dehydrating oven at different temperatures, so from 25 to 50 degrees, where I took them out and measured their mass loss periodically, which was essentially the mass lost as water through the leaves. And from here, I calculated the minimum conductance, or the G-min for each species. And then I compared these G-min at a standard 25 degrees, or the lowest temperature I measured between the species. And so that's what you can see here. So that's what you can see here, again, separated by subgenus, and looking at that overall trend across climate from PC1. And you can see that, as we expected, G-min tends to be lower in environments adapted, in warmer and drier climates where species have adapted to lose less water. And that makes sense because they can't necessarily afford to be losing heaps of water if there isn't much in their surrounding environment, or if they're exposed to warmer temperatures and transpiration rates are naturally higher. And species from eucalyptus, from the eucalyptus subgenus also tend to have a lower rate of water loss than those from the Symphyomertus subgenus, which goes along with what we saw in terms of cavitation vulnerability, where in Tasmania at least, eucalyptus species tended to have adapted into more warmer and drier regions than Symphyomertus species. And here again, we had one species that was a significant outlier in this relationship, and that was Eucalyptus gunnii down here, which lost a lot less water than we expected, given that it was from a relatively cool and wet climate. And so this dotted line, again, here represents the relationship among the Symphyomertus species, but this one without gunnii included. And so again, you can see there's a much stronger relationship between the other species and climate. And what's interesting about Eucalyptus gunnii in particular is that it was the most cavitation resistant species in my data set. And also Eucalyptus gunnii divaricata, which is the subspecies that I collected, is also known to be the most frost resistant or the most frost resistant and cold tolerant eucalypt species in general. So it's possible that there's some kind of adaptation in its cuticular structure that's preventing frost damage. And that's also worked towards minimizing water loss. But we didn't see this in any other species. And so that's mostly just speculation because it was a particularly tough species in terms of both traits. And so the reason we looked at several different temperatures for G-min, as you can see here, is because while it tends to start off relatively stable as the temperature increases, eventually there reaches a point where the cuticle begins to break down and becomes significantly more permeable, which leads to this increase in water loss that you can see here. And the point at which this happens is referred to as the phase transition temperature, or the TP. And the temperature that this happens at is something that varies between species, just like G-min in general. And so I looked at where that point occurred in each species and compared that TP. And we found pretty much, again, what we expected, that the species in warmer and drier climates tended to have higher TPs or could withstand higher temperatures before getting to that critical point of cuticular breakdown and water loss and increased water loss. And again, though each subgenus had a slightly different slope, the trend was moving in the same direction. And now an interesting point here that you can see is Eucalyptus dalrympleana, which had a TP of above 40 degrees, which is much higher than any of the other species. And the thing with that point is that while other species were sampled through winter and autumn, dalrympleana was actually sampled over the summer. So we suspect that its higher TP might have something to do with seasonal plasticity and a change in the cuticular wax structures throughout the year. But we don't have any other data to back this up since this was the only one collected in summer. So that's just a point of interest for now, but something to keep in mind as we're comparing all the species. And so those are just the Tassie species that I've looked at, where they show a bit of adaptation towards heat and drought tolerance, and also some interesting adaptation towards frost tolerance. But what I want to look at now is expanding this across the broader eucalypt phylogeny, at least in terms of drought and heat or drought and frost tolerance, if not these exact traits. And so, while I initially looked at just a small sample of Tassie species, you can see the range that eucalypts cover in terms of Australia's climate here in this figure that's been made up by Rachel Gallagher, representing the temperature and aridity across Australia. And you can see that this grey area represents all of the climatic niches that are covered within Australia. And the red circles represent where eucalypts fall along that range. So you can see that they cover most of the climatic variation in Australia. And so while I initially focused on a smaller section of the phylogeny as well, and the Tasmanian species within that, I'm now looking across the broader phylogeny, and where we can utilise this phylogenetic knowledge of such a large and diverse group of species to pinpoint where those trait transitions have occurred and relate that back to the climates that they've diverged into. And so to choose this larger number of species. We've chosen some species pairs across this phylogeny here. And some of these pairs we've chosen where a focal species has diverged into a cold climate exposed to freezing, and its comparative species has stayed in a relatively intermediate climate. And then other species pairs we've chosen for where a species has diverged into an arid climate. And again, its comparison species has stayed in a relatively intermediate climate. And so the species that I've started to look at now with that much larger group of eucalypts, we've chosen 106 that I can measure certain traits across and then compare that back to the climate data. And I've been lucky enough to make use of a huge eucalypt arboretum in Currency Creek run by Dean Nicole, where he has hundreds of eucalypt species growing all in the one common garden site, and where I've been able to sample from all of these eucalypts that I'm looking at. And so I'm looking at several traits to get an overview of their adaptive strategies for resisting water stress. So this is just a list of traits that I'm working through, either that I have samples for already or that I'm looking to measure in the future. And so it's a bit harder to take these physiological measurements on, as I was looking at earlier, on 106 species, so things like cavitation vulnerability. But one particularly interesting trait that Chris Blackman's been looking into recently is XLA, or the proportion of xylem area to leaf area. And he's found that XLA is a pretty good proxy for cavitation vulnerability. So it's something that we can use to gather similar information to what I've done using cameras and psychrometers, but instead just using a small section of the leaf PDL and some image analysis. And with images like these ones, that you can see here. And what's great about these is that we can use these images for things like vessel distribution as well, vessel diameter, vessel wall thickness. So we can get a lot of information about the xylem structure and the function in different climates through these sections. And so that's what I'm doing at the moment, expecting that xylem characteristics will be fairly correlated with climate, and particularly with drought and frost, which I'm focusing on, as they both seem to have big influences over how water stressed a tree is. And so we're expecting to see a higher XLA in species with more that are more cavitation resistant, so species that are exposed to frost or drought. And so that's what I'm working on at the moment, along with looking at some of those other traits and how they vary across the phylogeny. So just like to thank my supervisors and everyone else who's been involved in the project so far.